Bismillahirrahmanirrahim. Assalamualaikum everyone. Welcome to the stream of knowledge. Today we will learn about Staphylococcus epidermidis or Staph epidermidis. You can learn about Staphylococcus epidermidis by just knowing its name. Really? But how? Staph means grape-like cluster and coccus is the cocci or round-shaped bacteria. Mean round-shaped bacteria that are arranged in grape-like irregular cluster will be Staphylococcus. Epidermidis show the bacteria normally live on epidermis. We know epidermis is the superficial layer of the skin. What does this picture show? This picture shows us that Staph epidermidis is the gram-positive bacteria, cocci are round in shape and arranged in irregular grape-like clusters. Staph epidermidis is facultative anaerobe. Remember this. We already learned that all the staph species are catalase positive, mean they produce catalase enzyme, and this catalase will break down the hydrogen peroxidase to oxygen and water, and this oxygen appear as bubbles. We also learned that except Staph aureus, all the staph species are coagulase negative, and hence Staph epidermidis is coagulase negative, meaning it does not produce coagulase enzyme, and no plasma clotting will occur. And it shows white colonies. As you can see here, they show white colonies on culture media. But why? Because it does not produce any pigment as does Staph. And it shows no mannitol fermentation, means as you can see here there is mannitol fermentation and color has been changed, and there is no mannitol fermentation as does by Staph epidermidis. And it shows gamma hemolysis. Gamma hemolysis means no hemolysis at all. This is beta hemolysis and this is gamma hemolysis. This gamma hemolysis is shown by Staph epidermidis. We already learned that Staph epidermidis is the normal flora of our skin. Julie, if it's normal flora of skin, but how it can cause disease? And why we are studying this? It causes disease whenever it enters the body. But how it can enter the body? It enters the body when intravenous catheters or prosthetic implants occur. But how? For example, this is the intact skin and there is Staph epidermidis. But when the catheter or implants are entering the body, the bacteria will attach with them, and this bacteria now will enter in our body, and bacteremia will occur, as you can see here. For example, this is the artificial heart valve, for example, and then the staph will attach with it, and then when it will attach, it cause the pyogenic inflammation, and endocarditis will occur. But if it's artificial joint, as you can see here, while it's implanting, the bacteria will attach, as you can see here, and this bacteria will cause inflammation and cause arthritis. One of the main virulence factor of Staph epidermidis is its Glycocalyx, meaning it secretes the slime layer. As you can see here, it produces the Glycocalyx, and we already learned that the Glycocalyx form the biofilm. Although when many bacteria attach to this prosthetic implant, they will release their slime layer, or this slime layer will protect the bacteria from phagocytosis as they form a biofilm. It's just like a surface. It will cover the bacteria. Now no neutrophils will attack on it, no macrophages, no antibiotics, and it will cause continuously inflammation and infection. In this way we'll get infected. It normally causes endocarditis and arthritis and CSF shunt if the shunt is entered in the brain, and this shunt has epidermidis. It also causes sepsis in newborns. How we can diagnose that the infection is caused by Staph epidermidis? We just done the gram stain on the sample. If it is gram positive bacteria, then go for catalase test. If it is positive catalase test, then culture on it on agar. If it show white colonies and gamma hemolysis, then and no mannitol fermentation, it can be Staph epidermidis and coagulase negativity. Then main test which differentiate, it will be staph epidermidis. This is novobiocin, which is an antibiotic. Novobiocin sensitivity is done. The molar on molar Hinton plate, the novobiocin is attached for after lowering the bacteria staph epidermidis. If it is it show the clear area around the novobiocin, that will be staph epidermidis. And that was all about the staph. Can treat it by taking antibiotics and prevention. The skin should be disinfected before inserting the catheters and implants. That was all about staph epidermidis. If you really like the video, do like, subscribe, share, and comment. Goodbye, Allah Hafiz.